Hi, my name is Jay Jenkins from your biomed class. The two research papers I'm going to be discussing in regard to the history. Research on exon skipping technology is Gebsky and Dunkley. Um, Gebsky, whose title is Morpholio Antisense Oligonucleotide Induced Dystrophin Exon 23 Skipping in NDX mouse muscles. That paper revolves around like weekly injections of antisense oligonucleotides that were safe and tolerated in patients with Duchenne muscular dystrophy. There were no like adverse or serious effects reported. Researchers who undergo the study had a significant increase in dystrophin protein expression in the muscle fibers of treated patients, which suggested that the treatment may be effective in restoring normal muscle function. Because DMDs, when you have diminishing like fibers and tissue in the muscles, so you eventually have no functionality, making you be in a wheelchair. But yeah, so exon skipping in the MDX cells for Gebsky, it was seen that the MDX cells like could be induced by morpholino, which was like one of the antisense oligonucleotides. And it just showed advantageous properties explored by the outcomes, like the therapeutic outcomes of antisense oligonucleotides. Like for the morpholino sequences, it had like shorter sequences basically. So that was something that was like concluded. And what was unique about like Gebsky's, this is one of the like, I don't know if you can see it, but these are the sequences, but some of them were like changed around due to exon skipping. And there were outcomes of new sequences. What was unique about Gebsky's treatment was that like in Gebsky's treatment, she wanted to see if she could get her double-stranded gene, making a more stable and efficient exon skipping process that would then transmit into cells. There would be a shorter band in RNA. And yeah, that's pretty much like all for Gebsky. We can talk about Dunkley now. So for Dunkley, the title was Modification of Splicing in the Dystrophin Gene in Cultured MDX Muscle Cells by Antisense Oligonucleotides. What we learned was that if you're moving the exon, this could like lead to a point mutation. What a point mutation is, like in the article, it was actually a nonsense mutation, which was like discovered as a TAA sequence. So it was the second largest group of patients in a 14% Duchenne muscular dystrophy like chart. There were like myoblasts and MDX mice. There was also fluorescent molecules on the nucleotides that were like coded. The muscle cells and the MDX mice, they allow like this coding to, cross cell membranes. So yeah, the fluorescent then went inside the cells and into the nucleus. And after that, that's when RNA exon skipping and, the splicing took place. Fluorescent was put into nucleotides and coding like seeped into the MDX muscle cells with splicing. It's just like both had their own techniques. And yeah, we were like, I drew out the methods for Dunkley. So you can see like the MDX mice and the sequence, the cell membrane. Yeah. But that was my video of the methods, consequences, etc. for Gabsky and Dunkley.